Okay. Oh my God. I have a nightmarish, horrible headache. So it's a Friday afternoon. This morning we had a journal club and I recorded the journal club and I recorded the screen share, but the screen share messed up and I lost the recording. So all I have is essentially like a video of me in front of a green screen talking about random this paper. So I have to redo the screen grab or the screenshot and try to like write it out again, which is a pain because I hate drawing things more than once. I hate giving the same lecture more than once. I hate it. But I feel like this is a cool topic and I liked the paper and it was a challenge for me. So I felt like it would be worth it for me to come back in the afternoon, retry. So what we're going to have here is an interesting concept. It's going to be a reaction to my reaction of a CI paper. So this is a recursive reactionary paper reacting to my reaction to the paper. Here I have some ibuprofen and some migraine, etc. And I'll take a bunch of these babies. Oh, okay. And we'll see if I can do this. Test 1, 2, 3, test. It is functioning. Okay. I am John Beckmann, Professor John Beckmann, Dr. John Beckmann, the father of Sid, the transgenic mother of sin, your hostess with the mostess. Okay, I'm going to play the reaction. Let's get this baby going. Hopefully, I can sync the audio.
Maybe put like a saliva sample onto or something.
Otherwise, this would be a pain.
Onto and it would tell you if they were positive or not.
They have that technology for like malaria.
For what, for COVID or?
Yeah, they were training. I guess
it's going.
To
clearly going to be some edits here.
Detect if someone was infected with COVID.
Interesting. So that'd be phone.
So.
Also is allergic to bees.
Let's see if I can pull up the papers.
Okay,
here.
so we're gonna start. I'm recording this just so that you know. And the setup here is a little awkward, so I'm gonna do my best to do this well. This paper, okay. I sent out 2 papers. We will see if I get to both. I want to start with this 2019 paper. Variation in Wolbachia SID-B gene, but not SID-A, is associated with cytoplasmic incompatibility, mod phenotype diversity in Culex pipiens. Okay, and this is Mylene Wheels group. So let's talk about this paper. Okay, so of course good morning. Of course I love Culex.
I do love Culex.
And one of the reasons that Culex is a mosquito, Culex pipiens. Culex is particularly important for CI systems because some of the earliest discoveries like Levin and Marshall, who discovered and characterized CI, they were working in Culex. So Culex is a very famous historical example of CI. And beyond that, it's quite special. Levin was using it as a model to study speciation processes in mosquitoes. And the reason it's special is because, and I've drawn this before in prior lectures, what Levin originally did is he went around to different spots of Europe and he collected these isolates of Culex. Okay. And I think there were like 17. And he would do all these different crosses where he would cross X to Y or Z to A, etc., etc., etc. And in theory, they're all the same species, so they should be able to mate with each other and produce offspring. But what Levin found was that there was this very, very complicated crossing pattern where sometimes you would get offspring and sometimes you would not. Okay, so he called these the cytotypes. And this is what led to his discovery and coining the term of cytoplasmic incompatibility. So Culex is a very, very famous system. And he was right about this in a sense that it has always been this complex. It's always been, in some ways, I guess you could say it's a mess. It's very difficult to understand because there are so many different incompatibility types. And people have used this as a model for CI, and the thinking was that if we could understand how these types evolve, if we could understand what the process by which this happens, we could then understand the molecular variables that control cytoplasmic incompatibility. Okay, so the paper. Mylene Will's group has done a lot of work picking up on this. They're located in France, and let me see if I can scroll down here. They have followed up on this in a sense that they have many, many isolates of Culex, and with modern day sequencing technology and with our understanding of the Wolbachia genomes, there have been attempts to try to sequence many of the genomes that cause this, and then through comparative genomics, you should be able to understand what are in theory the CI factors. So you can imagine what you would do is you'd sequence a bunch of these Wolbachias, and if you have, there's actually in WPIP, fact check me if I'm wrong about this, but I believe I read that there are 5 WPIP types, but for the most part in most of these papers, they're studying 4. They're studying WPIP 1, 2, 3, and 4, okay? And these are, according to like molecular genetics, phylogenomics, different what they call WPIP types, okay? So again, the idea was let's sequence the 4 different types of Wolbachia and let's cross them together in different ways and let's do comparative genomics. And the rationale would be whatever is different between these 4 WPIP groups should in some way be responsible for the different abilities to mate with each other. That was the hypothesis. So that's the setup. Now there's a lot more context to these papers and why they're important. So another reason why so if we're thinking about like, why is this important? The complexity of this. So in Culex is very strange. If you compare this to CI and Drosophila systems, it's almost like it behaves differently. There's a couple differences between Culex systems versus Drosophila. And we have talked in the past about the Jiggins-Martinez paper, which described the life cycles of CI operons in Drosophila. And if you look at CI operons in Drosophila, what you tend to see is, right, there's a syntenic A and B, which is how we know it's an operon. And typically what you see is, in some cases, there's only one copy, like in WMEL, there's only one copy. In other cases of Drosophila, there are paralogs, paralogous operons. And what you tend to see in Drosophila is in many of these strains, there are disruptions. Here, usually on the N-terminus. In WYAC, there are some disruptions in the dub domain. And in Riverside, there are some disruptions on the N-terminus. By disruption, I usually mean single nucleotide polymorphism, or that induces a premature stop codon, or potentially a deletion, a single nucleotide deletion, which induces a frame shift and a premature stop codon. That's usually what's happening at the N-terminus. So in many cases in Drosophila, you see these kind of like pseudogenized Bs, and it's thought that they're probably non-functional. So the story in Drosophila of CI operons is one that you can visualize the death and the life cycles of these operons dying. QLEX is very different. You don't see the same pattern in QLEX, which is interesting. So in QLEX, the CI cytotypes are much more diverse. There's many, many more. And if you look at the CI operons, one of the big discoveries was that once we found this, once the CI operons were identified, this group, Myelin Wheels group, looked at the CI operons within these 4 different QLEX pipiens strains. And one of the hypotheses would be if the CI factors, in this case the SIDs, are responsible for CI. If that's true, in order to explain the vast complexity of CI in QLEX, there must be more than one set. Does that make sense? Like, you could never generate this type of complexity with just a single toxin and a single antidote. Does that make sense? So when we first, well, the toxin antidote was postulated very early. And this sort of like hypothesis was postulated, why is it not letting me draw? Was postulated very early in our understanding of CI, because everyone knew it kind of had to be like this. So when the CI operons were first discovered, there was sort of like a challenge and a skepticism that if you believe that CI operons are responsible for CI, you must prove this, because we won't believe you until you show that, like, we don't believe that one set can be responsible for this. And everybody kind of thought at the time, well, it's kind of easy to explain in theory. All you would envisage a situation where you get duplications of the operons. So you get prologous duplications, and then through just standard evolution and diversification, you could generate multiple CI cytotypes. Does that make sense?
Yes, it does
Okay.
make sense.
But it was Myelin Wheels group who proved this in 2018. So there's a prior paper, which I'm not discussing, in 2018, which is a Nature Communications paper, where they sequenced the CI operons and figured out that there were 4 copies, in some cases 6 copies, 4 to 6 copies in strains of WPIP. So that right there was kind of like a big discovery, in a sense that, okay, we can now understand this. You can understand how 6 different copies could give you vast complexities of crossing relationships in CI. So that was cool. And the way that they do this is, how do you think that you would sequence? Here's the problem. If you have 4 copies of SID, and they're all largely the same thing, right? They might only differ by a few SNPs. How are you going to get the sequences for them? What do you think?
Good question.
How would you sequence them?
How would you do that?
So the issue is, imagine you just run like a PCR. You want to sequence the gene. Your PCR primers are going to bind to all these, and you're probably going to get Sanger data that looks good, and then in one section you're going to get a whole bunch of like a mixture of stuff, right? Does this make sense? Mm-hmm. And so you're going to wonder, like, okay, like, well, what is the base at that spot? The only thing you can conclude is that there is a polymorphism in that location, okay? But you wouldn't know precisely what the sequence was. So it's difficult to get the sequence in that sense, but that data could at least show you that there were multiple sequences, okay? And
sorry, I was gonna say, in this case, would you resolve this through cloning?
So yes, that would be an important thing to do. You could clone them, and then when you clone them, you're only going to get 1 copy. Okay, but even an easier way to do it, which they do in the previous paper and in this paper, is you do they call it PCR restriction fragment length polymorphism. So if you get lucky and one of these polymorphisms is in the site of a restriction enzyme, you can do a PCR reaction and treat it with a restriction enzyme, and you're going to get weird bands that cannot be explained by just one sequence. And then you could, in theory, isolate those and maybe like clone those. So you can identify these different sequences through combinations of Sanger cloning and PCR restriction fragment length polymorphism analysis. So that's the context of like what they're doing to get these sequences. Okay, so let me pull up this figure from the paper, and actually let me go to my, can I go to my desktop? I want to pull up my actual notes on these figures, if you don't mind. Okay, so this first figure is kind of like is a lot going on here. In here, they're on the left side, they're describing the Drosophila melanogaster system, and they're pointing out to you how it's different. It's a monomeric monomorphism in a sense that there's only one operon. They're showing the A's by these Beckmann and the B's by these diamonds. Okay, so there's only one in melanogaster, and here they're showing, look, there's usually like 4 of these things in Culex pipiens. Okay, so you can already see the difference, and they're color coding these in a sense that now you get an idea that many of these things have different A's and different B's, so they are definitely like evolving, they're changing. Okay.
The dub stays the same.
Now let me go back and give more context. Again, this is a very nuanced CI paper, and so if you can understand all these discussions, you're on the cream of the crop in terms of CI nuance. Okay, so there are various models for CI. There's a 2x1 model and a toxin antidote model, and for the vast majority of thinking, they're kind of the same thing. A 2x1 can be interpreted as a TA, but essentially the gist is there are some people who think that both A and B are inducers in a sense that you somehow need A as a requirement for B to induce CI. And it could be 2 different scenarios. Could be one scenario where perhaps A helps B induce CI, and it could be another scenario where they're 2 separate toxins. A and B could be a different toxin. Okay, and in this case, oh, is that a question? Oh, sorry, okay. In this case, there could be the rescue in the 2x1 model is explained by, can be sufficiently explained just by A. So the 2x1 says you need both to induce, but you just need one to rescue. TA, classically, if you interpret it in its strictest sense, suggests that you just need B, which induces, and you just need A, which rescues. And they're sort of like a bimodal system, which sort of makes sense with a syntenic TA operon. Okay. Now, again, when I said 2x1 can fit within TA, like if you don't interpret TA too strictly, which I don't, there are certainly scenarios where you can imagine where an A has to somehow evolve, perhaps like a localizing function to B, perhaps they both have to get secreted, they're both expressed at the same time under the operon. Maybe it helps it localize and sort of like packages it and prevents it from being toxic at the wrong time, that's standard of toxin antidote operons. So there are some nuances between these, but they're not necessarily mutually exclusive. And there is data in multiple systems to support one or the other. The vast majority of the data supporting the 2x1 is only in Wolbachia from Drosophila melanogaster. Everything else suggests strict TA. So that is also the context of this, is that people are trying to understand, okay, what is the subtle actual truth to these models? And it could be a gray area. It could be like, one is perhaps an evolution towards the other, and you see mixtures of these in nature in different systems. Okay. just checking to make sure I didn't miss anything.
I predict we're going to see the subtle collapse.
Okay,
of
so
the
let's
2
look
by
at this
1.
figure. This thing. I'm gonna copy this. This was again a very nuanced paper for me to understand, even me. So I'm gonna try to, if I can zoom in on this.
Even me.
I'm gonna try to zoom in on this and explain to you the gist of like what they're doing. Okay. So what they're showing here are 2 key populations which are important in this Culex relationship. And the thing you need to understand this figure is you need to have read in detail the crosses that they're doing. So there's 3 key crosses that they're doing. Okay. So one cross is if you take a male from North Africa or actually I'm just going to do this slowly, one by one, because there's a couple crosses we need to talk about. Okay, so you take a male from North Africa, you cross it to a female from North Italy. This is compatible. Okay. You do the reciprocal cross, male, North Italy, cross to female, North Africa, compatible. This is an important understanding. Okay, so these 2 crossed together are mutually compatible. If that's true, let me write that out. If this is true, they must share an antidote. Does that make sense? That's the gist of what they're trying to say. So now look at here, they've visualized the repertoires of SIDS in these populations, and you can clearly see there is one that they all share, which is this green SID A alpha. All these populations have this. Okay, so there's a proposal that if I'm understanding this correctly, there's a proposal that this SID A and the actual full length, I think name of it is SID A4 for the WPIP4 alpha, is kind of like a master key for these lines. Okay.
I could be stretching the interpretation.
Now there's
of that
more
a little
crosses
bit.
that are important, so I'm going to erase this. Let me discuss now the other crosses that were done, which are informative. And I have to go this slow, so I apologize if this is too slow for people. Okay, so there's this line called Tunis. I'm assuming from Tunisia. They cross to females from Tunis, and there's 2 crosses: a male from North Africa and a male from North Italy. Okay, they do these crosses, and the outcome depends on what strain of WPIP4 is in that particular mosquito. In both cases, it depends. Let me don't be looking at this. Get rid of this. It depends. So this is when you see compatible or incompatible. There's different strains of WPIP4. Let's call this one like A, B. They probably have names for these. I don't know their coded names for these. So this is probably wrong in terms of their writing, but just for us, A and a B. Okay. And when they do these crosses, they get a different outcome depending on whether it's A or it's B. Okay. So now if you understand that, now the reciprocal thinking of what we just explained, to explain this, the inducer of CI must be different between the compatible and incompatible. Hopefully I'm getting this right.
Makes sense.
So then they look at, okay, in the incompatible strain, whatever's unique to this incompatible strain is probably like the inducer.
So it's this
Okay.
versus
So-
this.
They both have this. Hang on, let me look at my notes. This thing
What thing?
and this thing are not present up here. They're only in the bottom incompatible strain. Do you guys see that? Well, also this thing. Oh God, now I'm botching it.
No, I remember here. I
But
figured it
this
out.
one is also, so now it meant, remember there's two crosses.
Yep.
So whatever is explaining the incompatibility should be different between this and this and this and this. I see. So here's why we can cross
This
this one out,
and
is
this and
because
this.
this one is not different between these two strains.
Yep.
Make sense? So we can cross that one out.
Yep.
This
Not that.
one is not different between these strains.
This one is different in
Okay,
both.
so we can cross that one out. And what's left over is this SID-B alpha 2,
Yep.
which is different between both this one and this one. SID-B
This one is uniquely different.
A2. And SID-B B2. SID-B B2. So this is interesting that in order to explain the incompatibility, what has to be different would in theory be the inducer of CI, and they're seeing a difference in the SID-Bs. Okay?
Yes.
That suggests that SID-B might be just the inducer. Okay, let's look at the A's.
Look at the A's.
Let's look at the A's. Okay. This one we can cross out because it's up here. This one we can
Sure.
cross out because it's up here. So for every A between these two strains, every A is shared. Therefore, the A cannot explain the difference between this incompatible strain and this compatible strain. Does that make sense?
Fuck. I fucked
Okay,
that up.
so the conclusion here is
I'm gonna pause.
SID-B,
Oh my god, I fucked that up. Let me see if I can think through it myself. Okay, we're looking at the A's and we're questioning which A's are the same, which A's are different. The green A is shared on the bottom and the top. Therefore, the green A is not an inducer of CI. There's this SID-A, let's save that one for last. The blue SID-A here, delta, or is that epsilon? I can't see it. Delta or epsilon. The blue SID-A, that is shared in the North Italy population. So this North Italy population is unique because there are no SID-As that are different between this North Italy strain and this North Italy strain of these PIP4s, where one is incompatible and one is compatible. Therefore, you cannot explain the difference in modification or induction of CI based on a SID-A difference in the North Italy population. That is the gist, the big point of this paper. The SID, now in the North Africa population, I get a question on this later. There is this thing, which I'll talk about in a second. But I think that the quick summary of this is that there is no SID-A difference that can explain differences in incompatibility phenotypes, modification, the induction of CI between these two North Italy strains of WPIP4. Okay. Hit play.
can explain the difference, but SID-A cannot.
SID-B can explain
Therefore,
the difference.
if you're going to put
but
your
SID-A
money
can't.
on a 2x1 versus a TA, you probably put your money on the TA because in this case, the 2x1 cannot explain this difference. Does that make sense? That is like the entire gist of the paper.
What about the one from North Africa?
In this one, so it's interesting you say that, and that's one thing why they say. So you can tell, I can tell they really wanted to strongly make this conclusion, but I think the reviewers made them tone it down. And it's because of this North Africa population, because this one does have a different A. And so in theory, like the 2x1 could explain potentially this one,
North Africa,
but it can't
but
explain
not-
this one.
not
So if you're
Italy.
thinking about like what's the most parsimonious model,
It's the one that can explain
it's
both.
going to be the TA, which can explain both of these.
Yes, makes
So,
sense.
so I see what you're saying. It's good that you point that out, and I'm sure that the reviewers pointed that out. But that's sort of like the gist of this whole thing.
Got it.
Okay,
Makes
so
sense.
there's a couple controls that they do that are kind of important, which I suspect either the reviewers asked for or they were very intelligent in the sense that they included them. Can you guys think of any other rationales that might potentially mechanistically like explain some of these divergences? Like how could you still get different CI phenotypes even if they had the same repertoire of SIFs? Could you explain that? Like, is that a possibility? So some of the things that might are important to point out are like, what about like expression levels? So like if two strains, an A1 and an A2, even if they had the exact same repertoire, if this one was not expressing an A as much as this one, you might get a scenario where you might have an incompatibility. So there are like other like things to think about, and we have to be careful in the sense that there is a lot of nuance. We can't just look at differences to explain genetic differences to explain everything, because it could be an expression thing. And there's also, what was the other one? So in response to this sort of like question, they did do some QRT-PCR, and the QRT-PCR results did not essentially like overrule their conclusions. And there was one, I am trying to look for the other control. Oh, the other explanation is concentration or population of Wolbachia, which they might be expressed the same per Wolbachia, but if you have more Wolbachia in one, you're going to have more factors. So the other thing that would be important is make sure you check to make sure that the Wolbachia levels are the same. And again, they do these controls, and what the data that they find does not overturn their conclusions. So essentially what they conclude at the end of this is a cautious support of the toxin antidote model in favor of the 2x1. The 2x1 can't explain these patterns in QLEX, but the TA can. Okay, so let's go to the next paper.
All right, I'm not going to do the other paper that I presented was not as nearly as coherent as this. So I think I did a fairly decent job of summarizing this. My intent here was not to get everything. It's very difficult to present other people's work. This is not my work, so I apologize if I have misrepresented anything. I have just done my best. We're just doing a journal club discussing the paper. I thought it was a good paper. It's an interesting paper. It's a historical paper in a sense that looking at the CI, adding some new insights into 2x1 versus TA. It's about time I react to this paper. And I think I'm going to do this. I think I'm going to do a couple more reaction videos, certainly on CI in the near future. I think we're going to slowly start to see the subtle collapse of this idea that the 2x1 is a functional model of CI. It's looking to me like it's certainly not cross applicable to most of the CI operons. It might be true as a nuance condition of WMEL, but I don't think it's reflective of the actual general theme of CI mechanisms across all insects. And I think you're going to see that in the coming future as more and more papers and research come out. So I'm going to, in theory, do some more reactions to those papers as they come out, because I'm interested in 2x1 versus TA discussions. I think it'll be fun. Okay, so if you have any debates or any comments, leave them in the comments. We can we can have a discussion. We can write about it. If you have anything to say, if I mischaracterized anything, if I made any mistakes, write in the comments. If you thought it was enjoyable, if this was in any way useful to you, hit like, hit share, hit subscribe. Your hostess with the mostest, signing off.
Let the gate real time to die. I shall meet death head on. I shall meet death head on. Decker Kane, you've got to get out of here. Beware foul demons and beasts, there are many foes here. The time has come to cleanse my homeland. This monastery reeks with evil and corruption. Oh, I hate staining my hands with the blood of foul sorcerers. Eternal suffering would be too great for you, Diablo. Let the gate real time to die. I shall meet death head on. I shall meet death head on. Decker Kane, you've got to get out of here. Beware foul demons and beasts, there are many foes here. The prime evils are no more. The prime evils are